Welcome, everyone. This is our webinar on innovations in protein modeling, how physics is impacting industrial drug discovery, and it's brought to you by the Protein Society. My name is Liz Meyering. I'm the president of the Protein Society, and it's my great pleasure to introduce our webinar today on this hugely impactful topic in protein science. Before turning this over to our organizer, Jennifer Knight, I'm going to just make a few brief announcements. First, if you have suggestions for webinars, you can contact us at our website. We're always open to ideas for what the webinars could be about, and if you're interested in organizing one, we welcome you to contact us. Our next webinar is coming up on December 4th. It'll be on protein folding and misfolding in context. And I'd also like to invite you to join us for our next annual symposium at the end of June in 2025 in San Francisco. We have an outstanding program. We have loads of travel awards for young scientists. And again, you can find more information about this on our Society webpage. I'd also like to highlight our Society journal, Protein Science. This journal publishes a wide range of exciting protein science. And again, I encourage everyone here to publish your latest work there as well. And with that, I will turn to our main event. So I'd like to thank our organizer, Jennifer Knight, who envisioned this fantastic set of speakers. A bit of background first on Jen. She's a director at Schrodinger, Inc., based in New York City. She hails from Canada and obtained her undergrad and master's degree at Queen's University with Don Weaver. She then got her PhD in chemistry and chemical biology with Ron Levy at Rutgers University, and then did postdoctoral training with Charlie Brooks at the Scripps Research Institute and the University of Michigan. Jen joined the scientific development team at Schrodinger in 2012, and for the last 8 years has been a member of the Schrodinger Therapeutics group. She leads modeling teams employing the full spectrum of in silico strategies, including free energy calculations and machine learning approaches, and is a champion for workflow optimization and their implementation at scale to help drive drug discovery projects forward. She's an inventor on many patents and was modeling lead for the discovery of the Wee1 inhibitor, which we're going to hear about today, which is currently in clinical trials. I'd also like to thank our speakers, Marie Ledecq and Aysegul Ozen, for sharing their exciting advances with us today. And before I turn things over to Jen, I just want to give some logistics. So how are we going to do this? Each speaker will present for 30 minutes, roughly, including about a 5-minute question period. And after the talks, we'll have further questions in a discussion format among our speakers. So please take a moment now in your Zoom to find your Q&A tab, and you can type your questions into that tab at any time. Only the organizers see it, so you don't have to worry about it going to everyone in the meeting. When it's answered, it will move to the answered questions, and others can see it. It will be answered live. It may be said that it's answered live. We can't answer all of the questions during this time period, so that's why we'll revisit some at the end, but questions may also be answered in a typed format and be posted as well. And I think that covers the logistics. So with that, I'll turn it over to Jen, and I look forward to the talks.
All right. Well, thank you, Liz, so much for your introduction and for the invitation to organize this webinar. Back in the summer, as you and I were discussing the possibilities of this webinar, I really appreciated your encouragement and your kind of boundless enthusiasm for bringing these live from the field stories in the pharmaceutical industry to this series. So thank you so much. And also thank you to your team, Raluca and Shannon in the background that has made this a real pleasure to pull together. So the 3 presentations in today's webinar are focused on the common theme of physics-based modeling in live drug discovery projects. And I'm delighted to be joined today with Drs. Marie Ledecq from UCB Pharma and Aysegul Ozen sharing vignettes from her work at Nested Therapeutics. So as the term would suggest, in physics-based modeling, we're taking the system that we're interrogating and adopting a representation of it that can be expressed using mathematical equations that are based on physics. We're usually talking about the range of from quantum mechanical calculations, where we have full representation of the electrons, up to molecular mechanics simulations that are driven by these atomistic force fields, or even up to coarse grain type models. So given this scale in the pharmaceutical industry, we might run QM calculations to try to compare the reactivity of potential drug candidates, or perhaps optimize parameters that we could use in our force field to run the larger simulations, explore longer timescale events, for example, protein ligand binding that we're interested in. Now, projects in industry settings run the gamut in terms of the questions the physics-based calculations are trying to answer and how accurate or efficient the calculations need to be to make a material impact on the project. For example, when structurally enabling a new target or initiating a first-in-class drug discovery program, physics-based calculations might be leveraged to contribute to the fundamental understanding of the protein target itself. Whereas when we're deep into lead optimization, physics-based calculations might be focused on prioritizing compounds for synthesis, or perhaps understanding fundamental, like crystal packing properties that we might want to take into account for formulations. In another comparison, in fast-paced project in the lead optimization phase with extensive synthesis resources, physics-based predictions may be streamlined in order that we can help drive synthesis queue decisions. On the other hand, in another project, perhaps we're looking at undertaking a pretty complicated synthesis, maybe going from a linear compound to a macrocycle. Now we might want to run even more aggressive simulations to get that accuracy up to the highest it can be in order that we can best prioritize our synthesis, maybe more limited resources or longer time scale, impact longer time scales. So among the three presentations, you'll hear stories from projects in various stages of the drug discovery process and where there are a range of questions that the physics-based calculations are being asked to address. I think it's also interesting to note that these three presentations are coming out of pharmaceutical companies that range in size, from Nested Therapeutics with less than 50 employees, to Schrodinger with over 800 employees, and then an order of magnitude larger UCB Pharma with about 8,600 employees worldwide. And so that might impact the flavor of the stories that are being told, and we could get into that into the discussion as well. So I'd like to say a few words about our other two speakers, and then I'll dive into the first presentation. So Marie Ledecq obtained her PhD in chemical sciences at the University of Namur in Belgium. She studied with François Durand and Johan Wouters, and there gained expertise in molecular modeling and physical-chemical characterization of small molecules. Marie then joined UCB Pharma in Belgium in 2007 as a computational chemist and has acquired extensive experience in ligand and structure-based drug design. Through the years, she has contributed to numerous drug discovery projects, from hit identification to candidate selection phases. And Marie is particularly interested in multi-parameter optimization challenges, near and dear to my heart, where computational aided drug design can accelerate the drug discovery process. Her other interests include cooking, reading, and gardening, and she enjoys hiking in the forest and running with her oldest son, who's now 15. Aysegul Ozen comes to us from a chemical engineering background. She did her master's training at Boğaziçi Üniversitesi in Istanbul, and then did her PhD in biochemistry and computational structural biology at UMass Chan Medical School with Celia Schiffer. Aysegul then traveled across the US for a postdoc at Genentech, and then returned to the East Coast for positions at Pfizer, Blueprint Medicines, Scorpion Therapeutics, and Nested Therapeutics, where she served as senior director and head of computational chemistry. Aysegul has just started as senior director of computational chemistry at Odyssey Therapeutics, but we're delighted that she's able to join us today for the webinar and share about her experience at Nested. Outside of chemistry, Aysegul loves kayaking and hiking with her two Australian shepherds. So I'm delighted to be sharing this webinar alongside both of you, Marie and Aysegul, and I'm looking forward to your presentations and our discussion together. So I'll start in now with our first presentation. And the title here is Achieving Kinome-wide Selectivity in Drug Discovery Campaigns Through Free Energy Calculations. And this is part of our journey of our internal project team at Schrodinger in discovering novel, potent, and selective Wee1 inhibitors. So in this presentation, after a brief introduction to our Schrodinger Therapeutics group and our design goals for Wee1, I'll give an overview of our platform, where we use physics, and describe more fully the physics-based free energy perturbation, or FEP strategy that we used to optimize potency and to dial out interactions with a specific off-target. I'll then present how we use protein FEP simulations to systematically improve the selectivity profile of our series. The Schrodinger Therapeutics group uses a whole variety of computational methods to drive drug discovery programs forward. Within our collaborations with external companies, our efforts have resulted in quite a few DC nominations to date. We have 8 molecules in the clinic, 5 more in IND-enabling studies. And over the years, we have covered a variety of targets, from kinases and proteases to integrins and GPCRs. And we have compounds in hand and compounds that we're working on that we believe are poised to become best-in-class or receive first-in-class designations. We have also initiated our own wholly owned internal drug discovery programs. And this webinar is focused on our Wee1 program and the discovery of SGR-3515. Not the whole story, but one of the significant challenges that we needed to overcome along the way. So at a cellular level, Wee1 is a serine threonine kinase. It's a gatekeeper in the G2M cell cycle checkpoint. In the presence of errors or damage during DNA replication, cell cycle checkpoint proteins, including ATR, CHEK1, and Wee1, along with the repair machinery of the cell, work in concert to delay the cell cycle progression until sufficient repair of the damaged DNA can be achieved. So healthy cells repair damaged DNA often during the G1 arrest prior to DNA replication in S phase. However, cancer cells often have a deficient G1S checkpoint and therefore rely on these later checkpoints for their DNA repair. So thus, inhibiting Wee1 releases the cell cycle brakes and results in in cancer contexts, unsustainable levels of DNA damage, premature catastrophic mitosis, and tumor cell apoptosis. Now, publicly available Wee1 data from preclinical and clinical studies demonstrates a vast opportunity for inhibiting Wee1. For example, clinical trials with monotherapy of Wee1 inhibitors has demonstrated a 30% objective response rate, or this ORR, in uterine cancers, up to 50% ORR in ovarian cancer. And then in combination therapies that include Wee1 inhibitors, we've seen favorable outcomes in breast, head and neck, lung, pancreatic, and colorectal cancers. When we started our program at Schrodinger, there was one molecule in the clinic, AstraZeneca's AZD1775. And it was postulated that the interaction of 1775 with PLK1 was contributing off-target toxicity and was thus limiting its therapeutic index. So given the clinical validation that had been coming out and the concerns with AZD1775, our Wee1 program design goals were to discover a highly selective molecule with balanced drug-like properties and avoid drug-drug interactions and off-target effects. But I'd like to spend a couple of minutes here on the computational platform that we use in all of our drug discovery projects and then we brought to bear in this Wee1 program. So over the years, Schrodinger has invested heavily in the development of rigorous physics-based approaches for various stages and activities in the drug discovery process. And our vision is to be able to scan through vast amounts of chemical space, we could call it all synthesizable molecules, and with highly accurate models, be able to identify the single best molecule for a given target. Well, we're not necessarily there yet for predicting all of these properties, all these endpoints that we're interested in drug discovery at a highly accurate level across all projects and targets. But the vision is that when we have these models in place and we're growing the accuracy across the board, then this vision will become a reality and we'll have drug discovery processes that are faster, less expensive, and will have the fruit of bringing much higher quality compounds into the clinic. And we've already seen these kinds of efficiency gains in our drug discovery timelines, as we have high quality models often in our projects for potency, selectivity, solubility, and permeability, for example. And we're growing out the suite of physics-based approaches to be able to handle these other really challenging endpoints. So while this webinar is focused on physics-based calculation, we also use artificial intelligence or machine learning models throughout our workflows. And one of the places where we've seen a great benefit of the machine learning models is in this active learning approach. In this particular approach, what we do is it helps us profile a large enumerated library of design ideas. So usually what we will do is pick out a subset of compounds from this large library and run those compounds through our physics-based calculations. We then take the results from the physics-based calculations and use a machine learning model to train on those results, such that the machine learning model will recapitulate the physics-based models results. We use that machine learning model then to go back to the full library, profile all the compounds in there, and pull out the most promising compounds that we would then put into the physics-based calculations themselves. And we can go through this cycle a number of times to the point where we're satisfied that we have essentially culled all the good material out of this large library. And then based on these calculations, we can then prioritize the best of the best for synthesis and testing. And so this combination of the physics-based calculations, which have the high level of accuracy and the ability to extrapolate into novel chemical space, combined with the machine learning models that have the ability to profile large libraries efficiently, bringing sort of the best of both worlds together enables us to move really quickly and make significant strides in each round of synthesis. So one of our workhorse physics-based approaches include free energy methods. And this is one application of these where it's the prediction of a binding affinity for a design idea. So this is where we have a reference compound. We know the experimental binding affinity of this compound and the target we're interested in, and we know the binding mode of the target, the binding mode of the ligand in the target. And the question then is, well, what's the potency of a design idea? Should I make this? Should I not? And so what we do is we take advantage of the fact that free energy is a state function, and we construct then this thermodynamic cycle and take advantage of this cycle. And through simulations, we can then sort of fill in the gaps. So we do one simulation where we alchemically perturb from a reference compound to a design idea that would be in a congeneric series with the reference. Do this in the solvent environment, and we get a delta G free energy there. Then we do the comparable simulation, but now in the context of the binding pocket. And so now we have free energy estimates for the two vertical arms of the thermodynamic cycle. We know from experiment the binding affinity of the reference. And so this gives us then a really good estimate of the binding affinity for our design idea. And so when this physics-based approach is validated for a given target, meaning we know that the structure of the protein is appropriate for what we're trying to model, we know the binding mode of the series, this becomes a digital assay for our project teams, and compounds can then be prioritized reliably for synthesis based on these predictions. So we say computational or digital assay because the accuracy of this method is now approaching experimental levels for amenable programs. And here are two large-scale validation studies that we've published on showing FEP predictive accuracy across a large number of programs and a whole variety of targets. So this study on the left shows the average accuracy across a broad range of targets in both prospective and retrospective studies. And the study on the right it's a retrospective study of FEP errors compared with common experimental errors. And what we found here is that FEP errors are within 0.35 kcal per mole of the inherent experimental error, which makes these FEP predictions a powerful resource to drive decision-making in drug discovery programs. And so now back to the WEE1 project. So our early project goals were to identify novel chemical series that would be potent against WEE1 and selective over PLK1. And we used our computational platform, physics-based platform, to do this. We would take all of our design ideas and run those through a series of FEP simulations in the context of the WEE1 binding pocket and ensure that these design ideas would bind well to WEE1. In order to help dial out interactions with PLK1, we would then take the promising compounds from this first stage and run an explicit set of FEP simulations now in the context of the PLK1 binding pocket and ensure that design ideas would not bind well in PLK1. So compounds that would tick both of those boxes, potent against WEE1, not potent against PLK1, would then be candidates for synthesis. And we moved the project forward pretty quickly using this particular approach. And I'd like to just show you an example here of how we could move quickly, and this is and identify new chemical matter. And this was by using an auto designer core design approach, an automated workflow to enumerate and profile new scaffolds. So in this particular exercise, we took AZD1775. There was a nice publicly available crystal structure with AZD1775 bound to WEE1. So we used that structure. We had the solvent exposed region that of the molecule that we held fixed. And in the workflow, explored all sorts of different scaffolds, and then different decorations of the scaffolds, and then decorations of the decorated scaffolds, and explored a large amount of chemical space. The workflow then could filter the compounds to fit the properties that we had specified. It would fit good physical chemical properties of where we were at in the project, what characteristics we were trying to optimize for. We could look for fewer rotatable bonds, all sorts of things there. We can also ensure at this particular point from docking whether the design idea has a possibility of fitting within the WEE1 pocket. So all the designs that checked those boxes, and we ended up with 473,000 designs, those would then go into this an active learning workflow in an automated way as well. So now we would have this iterative cycle of FEP simulations, machine learning models to profile the rest of the database, and we would go through that cycle any number of times until we were satisfied that we had gotten the best of the best. And one of those new scaffolds that we had identified, and we had identified quite a few promising compounds from this workflow, was this novel 5,5 core. And indeed, the synthesized molecule indeed did have good potency against WEE1 and had completely dialed out PLK1 activity. So it was this sort of approach that enabled us to move really quickly in our project. So within 7 months, we had profiled billions of design ideas. We had would run 9,000 FEP calculations, and we'd synthesized 169 compounds. And now we had 5 chemical series, novel chemical series that we were really excited about. Had good potency against WEE1, certainly still room to optimize those, but a very good starting place, and had mostly dialed out PLK1 activity, if not completely dialed out. And so we sent representative compounds from each of these simulations, from each of these scaffolds to DiscoverX for testing in their ScanMax kinase panel. And we were completely blindsided when we got the results back. For those of you who are not familiar with this representation of the experimental data, this is the ScanMax output. Each of these disks represents results from one of our compounds. The red dot comes in if our compound was able to displace a control compound for a particular kinase. So each dot represents a different kinase that was hit, if you will, by our compound. And the magnitude of the red dot indicates the magnitude of the displacement of that control, or how much that kinase would be hit. And so we would expect a nice big dot for WEE1 and then maybe a couple smattering other red dots. But these results showed us that each of our scaffolds were completely promiscuous across the kinome. Now, we had already explored so much chemical space in the first 7 months, we didn't think we could just go back and do more of the same and just hope that the compounds would become more selective. And so we spent time examining what we might learn from these experiments and if there was any pattern in these otherwise messy and frankly very discouraging results. And in fact, we did find something. we found that different chemical scaffolds tended to interact with different subsets of kinases. One scaffold tended to hit these kinases, another scaffold would hit these kinases. And what we realized is that these subset of kinases were defined by the identity of their gatekeeper residue. And so with this observation, one of my colleagues, Anthony Clark, wondered aloud if protein FEP simulations might capture some of these patterns. Now, protein FEP uses a similar thermodynamic cycle as the ligand-based free energy calculations I showed earlier, but in this case, the compound remains the same, and what is perturbed is the sequence and the structure in the binding pocket itself. And so the perturbation is from the wild type gatekeeper to any of the other gatekeeper types that we want to explore. And from these FEP simulations, then we get an estimate of the binding of the compound to this mutant binding pocket. And what we found then is by running these FEP simulations, the FEP prediction depicted here on the x-axis that was predicted for 5 different mutant pockets lined up very well with what's depicted on the y-axis as a representation of the scan max output. And what we have on the y-axis is the percentages of kinases with that particular gatekeeper residue, the percentages of those kinases that were hit in the scan max panel. And so the protein FEP simulations were accurately able to identify which subset of kinases it would not interact with, and which subset of kinases our scaffold would tend to interact with. And so with the strength of this interaction, we went and profiled the rest of the compounds that we had sent to ScanMax, along with 2 literature compounds for which we had ScanMax data. And we saw this trend remain a really strong signal for protein FEP being able to recapitulate the kinome-wide activity of our compound. And so we decided to use protein FEP simulations prospectively to help us identify selective chemical material. So we had the same workflow at the beginning as before. We would still require design ideas to be potent against WEE1. And now compounds that would be predicted to be potent against WEE1, we would then confirm in the protein FEP simulations that indeed they would not be amenable in these other mutant binding pockets. We were able to move really quickly with this process. And so within 3 months, we profiled 445 million design ideas. 6,700 of those designs we ran through our WEE1 FEP simulations. 300 of those were progressed into the protein FEP simulations. And within these 3 months, we identified 42 designs for synthesis. And what was extremely encouraging is that we were seeing a signal in compounds becoming more selective, even in the first round of synthesis based on our profiling and prioritization with protein FEP. So what I'm showing here are a subpanel of 20 kinases that we gathered in-house, or rather at our CRO, so that we could get a quick signal, are we moving in the right direction, moving more selective or not? And so what we see for compound A1 is the compound that we had sent to DiscoverX, quite promiscuous even across these subset of 20 kinases. And in the first round of synthesis, we significantly reduced the interaction. And within the second round of synthesis, we're completely void of interactions with these kinases. And so out of the 42 compounds that we had prioritized for synthesis, a whopping 22 of them met the project potency and selectivity goals. And so this was an overwhelming success of the protein FEP strategy, and a very efficient way then for us designing out the promiscuity of our scaffold. So if we take a closer look at these 3 compounds that I showed on the previous slide, we now have the FEP predictions that I'm showing here, systematically becoming cleaner, if you will, against these different mutant pockets. And what was most encouraging is that when we sent compound A3 to DiscoverX, it is quite a bit cleaner in the ScanMax panel. This wasn't just a one-off. This is an example from scaffold B. Compound we sent originally quite promiscuous, and within a couple rounds of synthesis, we get a beautifully clean variation of the scaffold. We also use this method not just for cleaning up promiscuous scaffolds, but also for maintaining a good selectivity profile. And so we also use this as we were exploring new core changes. In this case, C1 is a literature compound. We knew it was quite clean. against the kinome, and we wanted to explore more changes within the core. So we did explore the changes, made sure that it would be predicted to be clean using our protein FEP simulations. Our potency wasn't as good right now because we were making some aggressive changes. And so the next round of profiling was to improve the potency against WEE1. So we now got it down to 1 nanomolar IC50 against WEE1, and we was predicted to be clean. And again, our ScanMax data confirms that it was clean against the kinome. And so we continued designing, profiling, and synthesizing compounds based on the FEP predictions and the protein FEP predictions. And not only did our compounds have great physical chemical properties and bind selectively to WEE1, but they were showing remarkable effect in reducing tumors in vivo. So I'd like to show you one experiment here. In this case, we have 3 cohorts of mice that had tumors implanted in them, and the tumors were allowed to grow. In the one cohort of mice, they didn't receive any treatment, and as expected, the tumor continued to grow. The other 2 cohorts that received either AstraZeneca's 1775 WEE1 inhibitor or our compound 8123 saw a decrease in the tumor size that was quite dramatic. Now, we stopped treatment after 26 days and continued to observe the mice and the tumor. And what we saw for the mice that had received AstraZeneca's 1775, after a couple of weeks, the tumor started to regrow and then was growing at such a rate that it looked like it had never received any treatment at all. By contrast, the mice that had received 8123 continued to see a reduction in their tumor size, and this bore out up to 4 months after we stopped dosing. And in fact, usually at the end of an experiment, we would do some analysis of the tumor tissues, and we didn't have enough tumor tissue left to be able to do the analysis. So this was kind of best case scenario. And so with this kind of profile, we're excited about the potential of our compounds to make a difference, not just in mice, but to make a real impact for patients in the clinic. And so additional profiling led to the discovery of SGR 3515, which is now advanced into phase 1 clinical trials. And so here I'm showing a comparison of SGR 3515 with its optimized selectivity and physical chemical properties alongside AZD 1775, which has since been discontinued, and Zentalis's compound, which is in phase 1 and phase 2 studies. And you can see the improved selectivity for SGR 3515. We also have no CYP3A4 inhibition and no CYP3A4 time-dependent inhibition. So we're very excited to have this compound being moved into clinical trials. And so without a doubt, the discovery of SGR 3515 was driven by our physics-based platform, our FEP and protein FEP simulations to be able to maintain or improve the potency and to improve or maintain our kinase selectivity. I haven't had a chance in the webinar to discuss other approaches that were important, but we use machine learning models and other molecular mechanics-based models and other quantum mechanical-based models to help balance metabolic stability and permeability, and also to reduce the CYP3A4 TDI liability. So through the lead optimization campaign, we used the variety of physics-based methods here to prioritize 300 compounds for synthesis, and we were able to systematically go through and move successfully to the identification of SGR 3515 as our development candidate, and actually now our clinical candidate. So it's a privilege to be able to share this part of the journey with you here on the webinar, and a privilege to work with such a talented, creative, and dedicated team at Schrodinger. So thank you to them, and thank you to you all for joining us in this webinar, and I'll be happy to take a couple of questions or two before Marie's presentation. And so I think Marie's...
Thank you.
Hey, there we are.
Thank you, Jennifer, for the nice presentation. I see here some questions, so maybe I can read some of these.
Sure.
So The first one is protein ligand binding is not purely in terms of thermodynamics. The kinetic constraints are equally important. How do you take into account this aspect of protein structure?
That's a great question. So looking at thermodynamics and kinetics there, and actually there's a nice tie-in with our We1 story, because we think that actually some of what we're seeing in the impact, the long-term impact that we're having, can also do with the long residency time that this scaffold has within within We1 itself. So that would be a kinetically driven event, and where we think we can have accumulation in the tumor because of that kinetic property, not just because of the favorable thermodynamics. So the balance of those two is certainly important to get right in the drug discovery programs. We do have some methods for trying to predict kinetics. We don't have, I'd say, the same level of accuracy right now that we enjoy for the thermodynamic properties. So that would still be, it's not our workhorse method yet. So we're probing that really primarily with looking at the effect that it would have experimentally first. That's when we would pick up the trends. But certainly once it becomes important, then we can start either bringing the simulations in or looking at specific experiments that could give us more insight.
Another question, maybe, if we have time. Is there a sweet spot with respect to kinase selectivity and promiscuity? For example, some promiscuity is important and perhaps necessary for killing cancer cells, but preserving normal cells. Any relationship studies done to understand therapeutic ratio versus promiscuity and selectivity?
Yeah, great question there. So it sounds like when is synergy, when are you hitting multiple targets and that's a good thing, and when is that problematic? So certainly with the extreme that we saw with our compounds that we got from the Discover X ScanMax panel, we know that that would kill the cells before it could do any be of any benefit. So we knew that that extreme is not clean enough to bring into even any in vivo kind of context. But some compounds can have a positive effect when you have multiple proteins that we're hitting. And in fact, this actually comes up in the We1 story as well, because we do believe that our compound is hitting, well, we know our compound is hitting MIT1, which has a synergistic effect with We1. We have a synthetic lethal relationship between We1 and MIT1. And so if you have point mutations in both of those, the cell is not viable. But if you have point mutations in one or the other, the cell can live. And so when our compounds are interacting with both We1 and MIT1, then we think that that's also contributing to the longer benefit that we're seeing from our compound. So there are sometimes, absolutely, when hitting multiple proteins, particularly if they're in the same pathway, can be really beneficial. So you're kind of preventing the cell's really brilliant kind of workarounds if one protein comes offline, as it were. The problem is, as we're trying in the biological context to understand how our compounds are working and what's actually bringing the benefit, then it gets murkier as we have it hitting multiple proteins. And then certainly for FDA approvals downstream, we have to show that we understand why our compound has the effect it's having because it's hitting certain proteins. So it's certainly nicer to have cleaner compounds, and nicer when we can understand when there are those synergistic effects that could be beneficial.
So I think that we can keep maybe the next question for the end of the webinar.
That sounds perfect. Who missed at the intro, Aysegul is at Odyssey Therapeutics. She's just days into her position as Senior Director of Computational Chemistry there. But I'm really grateful that she was able to continue to be presenting the webinar today from her work at Nested Therapeutics. So Aysegul, we see your screen beautifully, so over to you.
Thanks. Great. And I hope you can hear me as well.
Can hear you well. Thank you.
Perfect. Well, thank you. Well, I got some big shoes to fill going after Jen and Murray. They had both wonderful presentations. So I thought, I think, I mean, I wouldn't say it's now a standard, but a lot of companies in industry these days are using FEP to drive chemistry prioritization in different levels, and I think Jen's We Want story was a great showcase how you start with starting chemical matter and then drive it all the way to the clinical compound. What I wanted to do today is to give a little bit more examples of FEP impact across portfolio, across different stages of projects, beyond chemical prioritization. But I also wanted to talk a little bit about examples of different flavors of physics-based simulations to address some questions that ultimately can help, assay cascade formation and mechanistic questions, resistance predictions, and so on and so forth. So I'm currently not at Nested, as Jen mentioned. So this presentation has been anonymized, so I'm not going to tell you any target names or show necessarily chemical structures, but I'm hoping that it will still serve the purpose of giving you a sense of how we were able to use these type of approaches to impact different projects. So okay, maybe where I should start is at Nested, what was the need for computational chemistry? Like, what was it that we were trying to solve for here? Around 3 years ago, a little over 3 years ago, when Nested Therapeutics was formed, we had two front runner programs where we had some good chemical matter that they needed to be optimized for clinical compounds, and then those were the more later stage lead optimization type of programs, but also there was a need for supporting the fully computational new target identification platform, where essentially in a nutshell, we took all the cancer-associated genes, all cancer-associated mutations, we cataloged them, and then we mapped them on three-dimensional structure wherever there is available experimental structure, if not AlphaFold predicted structures, and then we clustered them, and then we predicted potential pockets, binding sites from those predicted structures, and then we basically cataloged, annotated all these potential new targets, novel targets. So that was a purely computational exercise that needed to be supported. So with that kind of background, you can imagine we needed to build the infrastructure and the computational chemistry capabilities to support all the way from target ID to hit finding, hit optimization. So your druggability assessment, structure prediction, cryptic pocket predictions, and then some of your more virtual screening, as well as some of your more advanced, sometimes more traditional CompChem support in later stages. So this was an example from the platform where we took this cancer-associated gene, row A, and we mapped the mutations in 3D structure, and then we pulled all independently determined structures from the public database, and then we just mapped potential pockets or potential binding sites in relation to these mutation sites. And then for more specific interest, mutants of interest, we actually did the mutation and then we ran long time scale simulations, and then looked for cryptic pocket openings that are specific for the mutant. And then eventually the goal is to come up with a targeting hypothesis where you can link your ligandability to drugability. You want to identify a site that can bind a small molecule that will ultimately have the right properties, physicochemical properties, to be administered as a drug. So this was an example that we've gone through computationally. And you can imagine certain parts of this was very high throughput. So this is an example. For example, for this particular protein, we had internally determined several crystal structures of different mutant variants of this protein. And we just implemented internally this algorithm from Greg Bowman, who's a professor at UPenn, his pocket miner. It's basically based on a training set. His algorithm has learned what are some of the local properties of a protein structure that's ultimately going to be more likely to open up a cryptic pocket. And then once you have that model, you can run it very quickly on existing crystal structures, and then you can sort of identify which residues are more likely to open a pocket. And then you can decide what you want to do afterwards with it. You can either select those for physics-based simulation, long time scale simulations to actually see the opening of that pocket, or you can actually say, I'm going to prioritize those targets for actual high throughput screening approaches. So this is one example. But then sometimes if you don't have the internal crystal structures, or for the point mutants, so sometimes it's a little bit difficult to predict from the static structure. So we've also implemented internally another algorithm that was published by Dorothy Kern earlier this year in Nature, and this is sort of a different flavor of AlphaFold, if you will. But by using subsampling of the MSA in AlphaFold, you can try to see, you can try to get at different subpopulations. You can try to get a sense of the conformational heterogeneity in different parts of the protein, which can hint you about where in the protein your cryptic pockets might open. But these are all relatively higher throughput and less, I would say, physics-based approaches. And then sometimes you just have to go to your physics-based simulations that are, and I think I saw one question, somebody was asking, what is physics-based simulations? I don't know if there's a consensus on that, but the way I think about physics-based simulations, the entire physics of the binding event is described in that simulation as much as possible. That includes the direct interactions, the waters, explicit solvents, and all that. So it's because of all the complexity, physics-based simulations tend to be a little bit more time-consuming and expensive. So we want to use it, especially in the context of a small company where you have limited resources. We want to be as practical as possible at where we want to apply those techniques and find the key questions that are really going to drive the programs forward. And I highlighted a few examples, which I'm going to walk through. So this is an example. For example, we started with this cancer-associated gene. We clustered the mutations. Cryptic pocket was predicted, but then we needed to characterize a little bit more the pocket dynamics, and I'm going to tell you more about that in the next slide. But then after this computational pocket characterization, we sort of, need some sort of experimental sanity check to sort of increase the confidence in the pockets that you find from your simulation. And in this particular case, we resort to HDX experiments. And then for virtual screening also, I'll tell you in a bit where it makes a difference to incorporate physics-based simulations and some more filtering that comes with those kind of predictions. So this is sort of a little bit more on cryptic pocket identification. I mean, I think if you're interested in cryptic pocket identification only for the purpose of prioritizing targets for high throughput screening, you're probably good. At the end of the second step, you need to detect your pockets, and you probably want to rank those pockets to get yourself a little bit more intuitive information compared to your benchmarks. How druggable how this protein is, how likely am I going to see any hits if I do a virtual screening. And for that, we use either long time scale simulations, and we run some sort of pocket detection, a binding site detection algorithm, sitemap, fpocket, whatever you use, over the trajectory, and then you have these densities that pop up. Or you can also resort to co-solvent simulations where you have these little organic probes that are water miscible in your simulation box, and then those as those little props spend more and more time at certain patches on your protein surface, that tells you there's a chance that a small molecule can also bind there or pick up some potency by interacting with that site. And then once you have those pockets detected, so you kind of want to know which pocket is more likely to be the winner. So the way, I mean, we've looked into this with quite a lot of different approaches, but then what we ended up doing for us that worked the best was ranking by using the druggability score, nonpolar volume, as well as the structural variability of the pocket lining residues. And then that sort of aligns well with some of the known pockets, pocket rankings from literature. But then if you, like after this step, if you push your project to virtual screening, then it becomes a little bit more complicated, because these pockets, these binding sites, they're cryptic, they're not very stable. That's what makes them cryptic. So they undergo a lot of different conformational fluctuations. And sometimes the pocket shapes that appear to be very nice, large, druggable, they tend to be high energy conformations, and they are short-lived because of that. And then you have a little bit smaller pocket openings, and they appear to be less druggable, but they're long-lived because proteins like to collapse on themselves. So when you're setting yourself up for success, so there's we found that the chances are better if you incorporate all of these in your virtual screening approaches, efforts to some extent. And like I said, once you have isolated those pockets, you have different ensembles. And then I didn't mention here, but we typically have done some pilot screens as well to sort of get a sense of what kind of compounds are we pulling into these pockets, and what's the docking score distributions and whatnot. But mostly these are first-in-class targets, meaning these are proteins or binding sites that were previously undrugged. So there's no actual known ligands. And there is a cost associated with screening very large compound collections virtually as well. So to make it a little bit more stepwise and computationally, from a cost-wise, computationally effective, we resorted to using co-solvent simulations to get a sense of the fingerprint of the pocket, if you will. So that would help us in the absence of a known ligand, those co-solvent probes, that helps us to get a sense of the shape as well as the some of the pharmacophoric features of the binding site. And then we move on to the receptor-based screening. And then at that step, I'll show you in the next slide, we benefited from incorporating some post-stability and FEP filtering. So this is an example where we screened a shallow flexible pocket. We screened over 1.4 billion compounds using active learning, as Jen described in her presentation. And when you screen billions of compounds, even your top score, like even if you look at your top hits, you're still looking at dozens of thousands of compounds. So you still need to keep filtering down. And in this particular case, we took two approaches. One was informed by our existing SAR. We said, okay, there's this particular substructure. We want to pull compounds that has that substructure, and we want to pick a diversity set from that substructure. And then in the second filtering scheme, we emphasize novelty. And then we ended up ordering about 35 compounds, and then we had about 6 compounds that showed 100 micromolar activity. So at that point, we went back to the drawing board and we said, okay, what if we took this 35 compounds that we ordered, and then we ran, instead of using just the docked poses and docking scores, what if we ran short MD simulations, 10 nanosecond, and then use a known weak active compound as our reference and calculate ligand RMSD as a form of as a proxy for binding post stability. Then what would happen if we put a cutoff for ourselves, ligand RMSD of 2 to 3 angstrom stability, then we would be filtering out around 25 to 40% of the false positives. And if you did that, not using 10 nanosecond, but if you keep cutting your simulation shorter down to 3 nanosecond, you're still able to identify unstable compounds in this pocket. So with that, now if you take it back to your filtering scheme and add this post-stability steps here, and also de-risking by FEP, your cluster representatives, then again, we ordered another set of 30 compounds, and then we had one more potent active. So definitely sort of pocket calibration of these sort of metrics based on the pocket that you're interested in. It seems to be helping with removing the false positives from the results. Okay, so this is another flavor of physics-based simulations, another example. This is more later stage. At some point within a congeneric series, we had 5 compounds that had a range of potency from 30 nanomolar to 1.5 micromolar-ish. And we wanted to understand the SAR here. What is the structural basis of the SAR? Here, so we use this method IFD-MD that's taking induced fit docking and as well as MD simulations. And then we predicted the binding poses for these 5 compounds. And what we saw here was there's these 2 solvent exposed loop regions that tend to be packed better with the more potent compounds. And just one simple metric to measure as a proxy for that packing, better van der Waals packing, is the distance between these 2 proline residues. And then for the weaker compounds, that packing is less optimal, so then you see a separation. So that works out, that these type of simple metrics work out very well for chemistry prioritization or incorporating as a design constraint, because it's easy to calculate, it's easy to understand conceptually, easy to design for. So we incorporated this as a constraint, and then the SAR further down, it tracked, and then we also confirmed this later on by crystal structures. Okay, so this is another example where we had, because we had gain confidence in this IFD-MD technology for this particular project, actually. We had a virtual screening hit that showed some weak activity, about 30 micromolar in a biochemical HTRF assay. But unfortunately, we did not have any orthogonal activity in DSF SPR assays. But the hit looked very interesting. And David Bellinger, the MedChem lead in this program, because we were able to show IFD-MD working very nicely and confirmed by crystallography before, he actually suggested, why don't you predict the binding pose for this particular hit, and then if the IFD-MD pose matches the pose that came from virtual screening, which is based on just rigid docking, then that's an orthogonal computational validation, and then we can take the risk and invest in making a few analogs because it could potentially be a backup series. And then when we did that, the poses matched, and then David was able to design a few compounds and improve the potency down to 4 micromolar. Then we were able to start seeing orthogonal activity in SPR, and then that the predicted structure was also confirmed later on with predicted binding pose confirmed with a crystal structure as well. So some of these targets, when sometimes we have these binders or we have these hits, but then we don't have necessarily a crystal structure, or we don't have a direct binding assay, but sometimes all we have is just a biochemical assay, or maybe sometimes a cell-based assay. But we have a predicted pocket, proposed binding pose. So in that case, we want to know that binding, is it actually productive? Is the binding set that you think you're binding, is it what's causing the functional readout? So in that type of situation, sort of predicting the FEP, but predicting the binding affinities with FEP, and then stacking with the biochemical assay. If you see a trend, then it's a very good indication very early on, typically in the project, that you have a productive binder and it's not just a silent binder, or it's not doing something funky in your assay. So that's very valuable early on when the projects decide what assays they want to invest to set up and their assay cascades and all that. Actually, this is another example of the co-solvent simulations, but this time it's not necessarily to identify a cryptic pocket per se, but here we have a weak binder and we have a predicted binding pose, and then we want to quickly evaluate if there's any hope to improve the potency for this compound. So then there's 4 vectors here that are shown, for example, and then when we ran co-solvent MD simulations, a clearly one co-solvent hotspot popped up in R1 vector, and that was the vector that we prioritized. And then you can see with FEP the potency increase with that perturbations on that vector, and then that's sort of again, going back to this small company with limited resources, like it's very important to gain time with these type of prioritizations. Otherwise, you can always, get to the same place regardless. Okay, so this is another example where we use FEP to ask a mechanistic question. So we had actually, it's our clinical compound at the moment, NSD628, but this is a non-degrading molecular glue that binds mostly to one kinase, and then it it just stabilizes the heterodimeric complex with another kinase. So the question was, is the stabilization, ternary complex stabilization, does it happen through direct interactions at the kinase-kinase interface, or is it that this compound is stabilizing a certain conformation of this kinase, and then now it's more prone to accepting the interacting kinase partner. So then when we ran FEP with the ternary complex versus the isolated protein, the results were very similar, as well as they were matching the cell-based results. So this was a good indication that the ternary complex stabilization is primarily driven by MEK conformational stabilization, as opposed to the direct interactions at the interface. That's also valuable to have that intuition when you think about the level of complexity of the assays that you need to set up for your assay cascade. Okay. So I think Jen mentioned protein FEP. So similar to ligand FEP, we can make these perturbations on the protein and ask a whole bunch of other questions. In this particular context, we wanted to see with our compound for this target, what other mutations in the binding site we can retain activity. So we took the excuse me, we took the cysteine mutant, that was the oncogenic mutant, and then we made all other 19 mutations, and we predicted, we ran FEP, and we predicted the binding affinity change. And of those 5 mutations that we think the compound was going to retain activity, there was only one, this serine, that was actually observed in only 3% of the patients. And then we suggested that to put in the cell data, and that showed not equipotency, but some potency. So based on this type of prediction and complemented or confirmed by the cell data, we propose that this compound, like we can add another 3% to our patient population and may be able to address that particular serine mutation as well. Another question we asked with protein FEP was, can we predict resistance hotspots? And for that, I guess you can think about resistance in this particular context as 3 pillars: the compound binding and thermodynamic thermostability, the mutations that are going to kick out compound binding. We don't want them to be thermally stabilizing the protein, otherwise they're not going to confer resistance. And computationally, by doing protein FEP, we can actually measure both impact of mutations on compound binding and thermodynamic thermostability. And also, we know that single nucleotide substitutions are more likely to occur than double and triple nucleotide substitutions. So taking all into account, if you want to predict resistance hotspots, what we did, we enumerated all possible mutations, and then we limited ourselves within 8 angstrom of the ligand, and then we filtered out all the double and triple codon mutations, and we were left with 206 single point mutations across 35 residues, and then we ran FEP on those mutations. And the red ones are your delta delta G is greater than it means you're losing potency, so you're conferring resistance, and then blue ones are tighter binding, you're conferring sensitivity. Now, some of these are resistance hotspots that are mostly red columns. Now, if you took those like top 10 of those mutations, and then you run your protein stability FEP, now it shows you all those are actually impairing protein stability. So there's a chance that they're gonna confer resistance. And then also if you take the ones that are the blue dots, meaning the drug sensitivity increasing mutations. If you study those a little bit more carefully, those are also the ones that are that can tell you about what you can improve about the ligand in that region of the protein to improve your ligand potency. So maybe I'll just pause here, but I think so as I mentioned, there's different flavors of physics-based approaches, and we're really limited by our imagination. There's so many questions in small molecule drug discovery that these type of simulations are helping us to address those questions. And with that, I would like to thank my team, the computational chemistry team, extremely talented folks. They're all moved on to their next adventures now, and the medicinal chemistry team, as well as the leadership. And I'm happy to take any questions.
Well, Aysegul, thank you so much for that presentation. I've always appreciated over the years the way that you think about, like, what is the problem that I need to address here? And let's get an elegant metric to be able to help our project team. So thank you for sharing these vignettes here.
Mm-hmm.
We've got a question that's come in around cryptic pockets. So the question is, how do cryptic pockets relate in shape and size to known cavities or packing defects in proteins? And what characteristics should they fulfill to qualify as druggable?
Well, I mean, I think based on the literature, I guess, and based on our experience, the cryptic, maybe should I, sorry, I'm trying to, okay. So based on our experience, I think the cryptic pockets by default, they're more flexible and they're cryptic, right? So but then that being said, I've been thinking about this a lot in the last, 3 years. Sometimes, like even if you take a kinase, for example, the ATP site is a pocket. There's no question around it. But if you look at a crystal structure of a small molecule bound kinase, and if you compare that with apo kinase structure of the same kinase, you're going to see a collapse in the ATP site. So it's very difficult to look at. I would say it's very easy to look at a structure computationally and say, well, there's a pocket there. I think that's going to be a pocket, but it's very difficult to predict exact shape that it's going to adopt if you have the right molecule. And then even if you predict that, finding that molecule is a whole different problem. But then in terms of druggability, it's really a balance of lipophilicity, which is a main potency driver in these type of pockets, and then some more specific interaction opportunities like hydrogen bonds. And if you have, a covalent handle nearby, that also helps for sure in these type of shallow or flexible pockets.
It almost sounds like sometimes the chicken and egg problem. If we had the ideal ligand, we could find the pocket, but we need the pocket to find the ideal ligand. And so how can we make those iterative steps forward? Yeah, thank you very much. And Marie, glad that you've joined us on screen here, and Liz as well, so we can move into the fuller discussion together.
I'm trying to stop sharing my screen, but it's stuck.
Well, we're not seeing the screen on right now, so.
Oh, okay, good. All right.
I think Luca has worked some magic in the background. But maybe I'll kick us off with one question together, and that is because in all three of our presentations, we've talked about these interdisciplinary, if you will, kind of collaborations, and was wondering what in your experience are sort of the best practices, or what really helps those collaborations to sing across functional domains? What do you bring? What do your team members bring that really enable these physics-based methods to make an impact on the projects? So Aysegul, you're nodding. Do you want to?
Yeah. I mean, I think physics-based approaches are, I mean, by default, I mean, I guess they're a little bit newer. So they're a little bit unknown to non-computational scientists. So I have found it very useful to actually take my time and sit with people and explain them what does this do, what can it do, what has it done in the past, pull examples, like some showcases, success stories, but also be very frank about the limitations, and have sort of like an open and honest conversation, and keeping it on the project goals. What do we need to achieve? That's what I have found very valuable.
Marie, how about you?
No. I fully agree with you, Aysegul. I think the secret is probably a good communication, but it's difficult to summarize what is a good communication. It's always nice to sit with people and speak directly with them, and also it's always nice to share our expertise and enthusiasm about some new technologies, and also see how these different methods can be orthogonal with each other and better answer the question at the end. So all these things are useful to demonstrate the utility of working together. But I'm absolutely convinced that it's really key to work in a multidisciplinary team, especially as a computational chemist. I cannot do anything without the experiment anyway. So then I need to discuss with people. And I agree with you, Aysegul, it's absolutely key to meet the expectation of people. So really understand the question, but also make them understand the limitation of the method that we are using, and have an honest and frank discussion about this. But this is the key, one of the key for success, I think.
Mm-hmm. Yeah, totally agree. Liz?
Yeah, I have a comment as a non-computational person, but collaborating for many years with computational people. So it's amazing to me, truly, looking at this, how far we've come in terms of over the trajectory of my career. And, things go in waves. And I think your points about communication are perfect because we have to learn the language. So things are becoming more accessible, so we can talk to each other and understand what each other are saying. Things are becoming more accessible. More and more people are using computation all the time. I guess, can you comment on where you think things are now in terms of the role of computation and where you see it going? I mean, it's hard, you don't have a crystal ball, but some things we can see in the nearer term, perhaps. And then there's, what next?
Yeah, it's a good question. In fact, I have seen also the evolution over the years of this kind of relationship. So at the beginning, I'm speaking about 20 years ago or something like that. It was more about believing or not into computational methods, like believing in God. So it's absolutely. So we are going outside this kind of mindset, and now especially in a pharma such as UCB, people slowly understand that there is a real need to change the mindset, especially when you design new compounds, to take better decisions. We have such an amount of data that is available. It's a pity not to capitalize on this data. And to capitalize on this data, we need the models, and we need to have accurate predictions. so with the limitation of accuracy, etc. But then people are using this information to take the best informed decision as possible, you know? So this is really changing, and I see that it's a more recent change. No, I'm speaking about the design, for example, designing new drugs. No, more and more the design is driven by the predictions, because people have access to the model, but it requires a lot of different conditions. So the first is a good communication, it's a kind of learning process, but also we need to make all these predictions easily accessible. Because if people are asking us a question and then we come back 3 months later, then it's too late. So, you know, we need to have all these criteria together. But I see the evolution, and probably in the future we will take more and more the decision based on the prediction that are based on all the data that we are accumulating.
I think that's a great question, Liz. And I was thinking about how we're able to profile now so many more design ideas at a time, and where that challenges kind of the paradigm of, well, let's see what the SAR is, and we'll infer from that the next decision. Because that's fine when you have 20 compounds, maybe 30, but when you're dealing with billions, then I can't hold that all in my head. So how do we translate what we've scanned through into some concrete, digestible suggestions for the project team in a way that is meaningful in terms of, like, there's a structural connection to it, but we're not limited by that. In some of our project teams, we might do this long enumeration, show designs to the team, and they'll be like, well, is the methyl really important? We're like, well, we just changed half of the molecule. We can go back and see if there's a methyl to ethyl hydrogen, and we can go back and say that, yeah, the workflow said that you need the methyl there, but we didn't necessarily have all that data right in front of us to show that it was necessary. So, so I think figuring out as project teams, how do we get that information in front of the team members? But also, how do we change some of the conversation too, so that it's not like, oh, we need to in this round do our ethyl, methyl, propyl, and synthesize all of those. Can we trust the calculation to move us in the right direction for this design round, and trust if our DC is in that enumeration that we somehow missed, we'll catch it a couple rounds from now. So it's okay. We don't have to fear that we've left one behind. So.
Yeah, I also agree. I mean, I think there's a lot of competition these days. Everyone wants to be the first one to get to the finish line. So everyone is interested in reducing their design cycles and number of compounds that they design every cycle. And computation is, I think, I mean, it's getting better and better. I think there's also sometimes, you know, there's some hypes around certain things. I think over time they get corrected, I feel. My personal opinion, like when AlphaFold came out, like that was a hype, right? Like we're never not going to solve any crystal structures of a small molecule because there's now AlphaFold, right? Like there's always going to be a need for collecting very key complementary piece of experimental data. But at the same time, also there used to be some resistance. Like I have heard years ago, like maybe more than 10 years ago, oh, you can never use FEP in actual drug discovery projects. It's so expensive, so time consuming. You need dedicated, I mean, yes, you might need dedicated people, but now a lot of people are using, right? It's not just Schrodinger who's able to do this, right? So I think it's, you know, it's getting more and more involved in small molecule drug discovery, I think.
Well, I guess, so I'll put it out there, just we're heading towards the close here, but right, hype now is around AI approaches and how they work with physics. So we've heard some about this, but it's you know, I think there isn't, I don't know, I'm curious on your perspective on that. forward? Like I think we need it all. That's my guess.
I'm a use it all and see how it's performing on our project. So we still see limitations in terms of the transferability, like a machine learning model that will solve it all. I'm quite skeptical of that anytime soon. But the active learning work that we do, physics-based calculation, to answer a specific question, train a machine learning model on that, the power of that, like I've seen that over and over, that it is amazingly efficient for helping us get to the right compounds quickly. So I will never trade that in. And the methods are getting better and better as that fundamental methodology develops. But now we're able to tackle like really more challenging endpoints. So clearance, bioavailability, things that are hard to have a really like an elegant physics-based model on, because if you're needing essentially to sample all the cellular processes, different organs, I mean, that's very challenging. So machine learning models will come in, I think, more there. But I still have the skepticism of what are the global models claiming, and will that pan out? But Marie or Aysegul?
Yeah, I also go ahead,
No,
Marie.
please go ahead. Please go ahead, Aisha.
Yeah, I mean, I completely agree with Jen. I think there's always, I mean, I'm always going to use machine learning and AI algorithms. If there's a good data set, I think data set is the key there. And also, sometimes even a simple regression model is very predictive if you have the right data set. So from a very practical drug hunter perspective, I'm always looking for the simplest approach that's going to solve the problem. But some problems are getting more and more complicated. And as we get more data to describe those problems, I think these approaches are going to be very, very valuable.
Yeah. I agree, and I think that this AI approach probably will be in the future one of the tools that we will have in our toolbox to solve the complex drug design, drug discovery process, you know? So at the end, it's also algorithm. It's exactly all the methods that we are using in computational chemistry, and it's relying on the data. And again, as you have said, the quality of data will probably influence the quality of the model at the end. So,
And my
yeah,
hope for machine learning. Yeah. Oh no, go ahead, Marie.
No. I'm just saying, okay, AI. There is a kind of hype to say that we are doing AI. We are using maybe machine learning algorithms, or active learning algorithms, or even generative AI, but it will be part of the toolbox in the future. That's it's not just, it's fantastic methodologies, but it will be part of our toolbox anyway.
And if I had a key wish for the methodology development for machine learning, is to have a good metric for saying, are we within the domain of applicability, or are we not? So we've got it feels like right now, like maybe some estimate of that, maybe more common sense is applied, but to have like a rigorous number on that, I would love that.
Yeah, that's true. And also, I think it's important to have some tools in place to evaluate the performance of the model, to be sure that the predictions that we are using are accurate enough.
Yeah.
So, yeah, and this is something that we are doing that more and more, and at UCB we are trying to monitor carefully the performance of each model that we're using prospectively. But this is absolutely key also to convince people to continue using this model. So there is also a part of communication there.
Mm-hmm. Yeah, the versioning control on those, so that we can look at what were the numbers on which we made the prioritization decisions, and
exactly.
Those bearing out. Yeah. And then how does the quality evolve over the course of the project? For sure. Yeah.
Well, I mean, I would like to thank you all again, because I think this was just an absolutely inspiring view of where things have come to in this area, and I am very optimistic about the future. I was that grad student who said, "Oh, we can predict something. Can we say this and that, right?" And then it's a very different answer now. And so I thought we had just a beautiful scope of work here presented, and thank you all so much for doing this.
For having
Thank you,
us.
Liz, too. Yeah, great to be here together. So.
Okay, and thank you to the audience. And this presentation will be on YouTube. So yeah, I wish everyone a beautiful day, evening, wherever you're at.
Thank you.
Thank you. Bye-bye.
Bye-bye.